another lecture of the course Aquaculture. Today we are going to discuss about farming of pearl oyster, Pinctada fucata. In the first lecture today, uh, we shall be discussing about the biology of Pinctada fucata, the pearl oyster. Pinctada fucata, the pearl oyster, is widely known for producing very good quality of pearls worldwide, and these pearls are very popular in jewelry industry. This is the classification which is shown here, that it belongs to bivalvia of mollusca, and genus is Pinctada and fucata. Pinctada fucata, or the pearl oyster, is widely distributed in Indo-Pacific region, from the Red Sea, Pacific Gulf, India, China, Korea, Japan, Western Pacific Ocean, widely distributed. In India, it can be found both on east and west coast. Um, the Pinctada fucata is distributed on east coast, Gulf of Mannar and Palk Bay, Tamil Nadu and Andhra Pradesh, Gulf of Kutch on west coast, Karnataka, Kerala. Morphology of Pinctada fucata: the outer shell is reddish or yellowish brown. There are radial rays which are lighter in color than the shell. Nacreous layer thick and has golden metallic luster. Long hinge only 0.85 times the maximum width of the shell. Left valve is deeper than the right. Both valves contain hinge teeth. The common features which we notice in case of Pinctada fucata, these things are shown here. Also, you can very well see the hinge, the byssus threads, as well as the umbo, ear-like processes, finger-like projections, shell valve of Pinctada fucata. This is a sedentary animal that we all know about. Such kind of mollusks occur in shallow littoral zone, often inhabiting closed inlets or bays, in enriched with fresh water or terrestrial enriched water, right? Attached to the hard substratum by its byssus threads. In the diagram I have shown you the byssus threads, which are very useful in attaching to the substratum. What are these substrates where they really attach? Corals, sand grates, rocks, and other submerged objects they can attach to. Occurs in large numbers on the submerged rocky substrata, which are often referred to as pars. These pars are found in the Gulf of Mannar in India, if we are talking about. Otherwise, they are distributed in many parts of the world, and especially in Japan, um, they are being utilized for wonderful pearl farming practice. In India also we are doing that, but Japan has been producing pearls from these Pinctada fucata for very long. Found in Gulf of Mannar at a depth of 12 to 25 meter off the coast along a stretch of 70 kilometer. So this Gulf of Mannar is considered very rich in terms of Pinctada fucata distribution. It is also found, it not uh, comparable to that extent, but in Gulf of Kutch as stray individuals on intertidal reefs. We often refer to that as khadas. So, Gulf of Mannar, they are distributed widely over a large area, and in Gulf of Kutch also they have been reported. They are also found in Gulf of Kutch. In Palk Bay, it appears occasionally on loose sandy substrate clinging to submerged items in coastal seas. Typically, they recline on the right valve at 40° to the surface of attachment, of course, with the help of plane of commissure. Food and feeding habit. These animals, bivalves, mollusks, we all know that they are filter feeder, and major food of these organisms include infusorians, foraminiferans, and radiolarians. However, Pinctada fucata has been found to prefer certain brown microalgae more than the other foods, such as Isochrysis galbana. This is considered one of the best food items of Pinctada fucata. Other food items, minute embryos they can feed on larvae of a different kind of animals. Algal filaments they can feed upon. They also feed on spicules of alionarians belonging to Nidaria, that and sponges. So a variety of food items they can feed upon, but it depends on in which region we are having these um, animals, and depending on the availability of the food in that region, you can get some variation in their food and feeding habit. That kind of thing must be noted. Now, coming to reproductive biology of Pinctada, sexes are separate, but hermaphrodite conditions have also been reported. Males and females are, in different individuals, but many a times you can notice that they change sexes from male. Pre-spawning, there may be dominance of one sex, or post-spawning there may be dominance of another sex. So that kind of condition has also been noticed in case of Pinctada fucata. Usually age at first maturity is 2 years. This is very important to understand when we are collecting the Pinctada fucata. First, you will have to assure that you are getting the brooder, or the Pinctada for breeding of the right age. So, never go for attempting breeding program for the Pinctada with less than 2 years of age. So 2 years of age, that is the age at first maturity. There's fast growing individuals. If the growth is fast, then they can attain maturity even in 1 year. In some cases, if growth is fast, you will get to see the age and growth data with relation to size. So if by that context, if you get to see that these are showing faster growth rate, then even 1 year individual may also show maturity. Slow growing individuals attain maturity maybe in 3 years. So the range is 1 to 3 years. Usually, in general, 2 years is considered age at maturity. So depending on the conditions, depending on the region, you can select the suitable Pinctada fucata for breeding. Spawning season in India, multiple spawning has been reported. Maturity here it is written in 7 to 8 months. So there is a lot of variation in maturity condition according to the conditions of that region. We all know that a number of biotic and abiotic factors have a strong influence on the reproductive biology of aquatic animals. So depending on the conditions, you can have, first you will have to ensure that the animal which you are selecting for breeding, that is mature, whether it is 2 years of age, or less than 2 years, or more than 2 years. The maturity condition has to be checked before proceeding further. Two peak spawning seasons in a year have been observed: June to September, and November to December. So these two main spawning seasons of Pinctada fucata have been reported here. Whereas in case of Japan, spawning season is in June to July. That also corresponds to one of the spawning seasons that we are having, June to September. In Red Sea, winter to spring, that time they can breed. Exhibits protandry, males mature first. That kind of condition is noticed in case of Pinctada fucata. In fact, maybe I shall be explaining you regarding how the sexes influence the quality of the pearl. Even either it is developing in male or female, that also has some kind of influence on the quality of pearl. Gonads are paired, and gonads form a thick envelope covering the digestive gland. So, sexual changes, as I discussed earlier, are observed between different spawning seasons. Uh, the sex composition of the population of one stock from one region or population from one region, you may get that ratio changing, male to female ratio changing in different spawning seasons. Sometimes pre-spawning and post-spawning sex ratio becomes different, and that is one of the peculiar features that Pinctada fucata natural population shows. At low magnification, male and female gonads are not differentiated. It is very difficult to differentiate the male and female gonads in case of Pinctada fucata if you are not careful enough, if you are not observing them properly. Male gonad uh, appears pale yellow, female gonad appears deep yellow. The mature gametes pass into tubules that converge into two vessels, one on each side of the visceral mass, that open immediately dorsal to the renal apertures. That's how the gametes pass. The 5 stages of sexual maturity, which is noticed in case of Pinctada fucata, is being given here in the following stages. Number 1 is stage 1. You can say that as inactive stage or resting stage or spent stage. Sexes, although spent stage has been discussed separately, but this stage is also considered as inactive, resting, or spent. That is very similar to uh, you know spent stage. Sexes can hardly be distinguished in this inactive stage. When gonads start to develop and mature, the transparent nature of the resting gonad is lost and it becomes distinguished from other visceral masses. Otherwise, before this, it is very difficult to identify where is the gonad, how to distinguish it from the rest of the visceral masses. So that becomes start to be discriminated at stage 2, that is maturing stage or developing stage. And the transparent nature of that resting gonad is you know lost. Stage 3, mature stage, the gonad spreads on to most of the visceral tissues. It is mostly yellowish cream in color. So by now, when Pinctada fucata becomes mature, it becomes quite visible that gonads are maturing, and you can easily identify even by their color. Then comes stage 4, partially spawned. The gonads become loose in consistency when partial spawning has already occurred, and the visceral epithelium becomes dull. Right? So gonad becomes loose in consistency, and the visceral epithelium becomes dull. So that is the characteristic of stage 4. Whereas in stage 5, um, when this is a spent stage, when spawning has already occurred, when the gametes have been released. So that is why the name is spent stage. This spent stage or this kind of classification you will also see in case of fishes also. The gonads shrink further with a few leftover gametes in the lumen of the follicles. So after spawning has occurred, the stage of gonadal development is known as spent stage. So these are major 5 stages which are described for Pinctada fucata gonadal maturation. Now, have a look on life cycle of Pinctada fucata. The mature oysters release their eggs and sperms through the paired gonoducts, which ends in the genital opening located at the anterior ends of gills. The eggs and sperms are released in the water and fertilization occurs. External fertilization occurs, so the gametes from both male and female, they are released, and then external fertilization occurs. The fertilized eggs settles down and pass through different developmental stages. When the egg is fertilized, it settles down and then development process starts. What are different developmental stages in the life cycle of Pinctada fucata? Some of the very common stages that you must be knowing: blastula stage, followed by uh, gastrula, then trochophore, then veliger, umbo, eye spot, pediveliger, plantigrade, and spat. So these are the developmental stages that are described for Pinctada fucata life cycle. Here it is shown diagrammatically, the oyster life cycle, pearl oyster life cycle. Eggs are released, gamete from the male and gamete from the female. Fertilization occurs. Here fertilized eggs are shown. Now development starts, and approximately after 2 weeks we get these stages completed, free swimming larvae. Now after this, spat attached to shell. After that, they need some hard substratum to attach upon, and then when they attach, then after 2 to 3 years they develop into either male or female adults. So that is the life cycle of Pinctada fucata. Blastula stage is characterized by two-celled stage formed after 45 minutes, about 45 minutes of fertilization. Macromere and micromere are formed, and then micromere do not divide, do not show any division. Micromere divides repeatedly and forms 64-celled stage, which we call as marula. Marula has cilium. This is an important development, and comes to the surface because of this, exhibiting phototrophism. This marula stage starts showing phototrophism, and because of cilia it can come to the surface. Reorientation of cells takes place at this stage and forms blastocele and blastopore. This stage is blastula and it forms in about 5 hours. Then comes the next stage, gastrula stage. This stage is reached in about 7 hours after fertilization. These durations are usually described after fertilization. Dermal layers and archenteron are formed. Then comes the next stage, trochophore. This stage develops a long single flagellum and preoral tuft of cilia. Long single flagellum and preoral tuft of cilia at apical side and postoral tuft at the rear side. So oral tufts develop and long single flagellum also develops at trochophore stage. This is a diagram where you can see that how embryonic development of Pinctada fucata progresses. Diagram has been taken from one of the publications available. Here in A, you can see how sperms look like in case of Pinctada, and in case of B, here eggs are shown. C shows single sperm on egg surface. In case of C, here we are describing single sperm on egg surface. Then D shows embryos after first division. D, this one. First division has occurred, then they look like this. Then we have embryos after second division, represented here, shown here in E. Then F diagram that shows embryo after third division. After this, we have G, which represents marula. You can see here very clearly the peculiar marula stage. Then blastula and followed by gastrula, the characteristics that we have discussed in the previous slides. Next stage of development comes, that is veliger. This stage assumes a D shape and secretes embryonic shell material. So from the veliger stage, embryonic shell material is secreted, and the peculiar shape, D shape, starts developing in the veliger. Now, flagellum and cilia disappear. So, veliger stage is characterized by disappearance of flagellum and cilia, and velum is formed. The time taken is about 20 hours. It is followed by the next stage, which we call as umbo stage. Addition of shell material in umbo stage also, and it occurs on 10 to 12 day, the addition of shell material. Early shell formation in Pinctada fucata, the characteristic that we have been discussing in the previous slides. A. Early trochophore. B. Trochophore displaying initial SFI. SFI is shell field invagination. So, how the shell formation is occurring in Pinctada fucata, that is shown. In A stage, trochophore stage is shown, followed by trochophore displaying shell field invagination, initial shell field invagination in B diagram. In C diagram, trochophore shell, trochophore with elongated SFI, this one, elongated shell for field invagination, shell field invagination elongated in case of C. Then in D, we have early shell material. D, we have early shell material on either side of the narrow crevice that will form the hinge. It is followed by E diagram, where shell extended over the trochophore surface to form a saddle shape. So, a peculiar shape started to form here, that has been represented in E diagram. F, developing shell and flat surface of hinge region. In case of here, the developing shell and flat surface of hinge region is shown. In G, we have a spread of shell material over the surface of heart shaped trochophore. So, how the shell material is progressing, how the shell is forming in Pinctada, this has been very beautifully shown in these diagrams. Then lateral compression of trochophore in H, it is quite visible now, and I shows early D-shaped stage. The peculiar shape is also started to take up. Followed after that stage, now we have progressed to the next stage, that is eye spot. Eye spot develops because of the characteristic. You can understand. Eye spot develops at the base of foot bud on day 15. These timelines are also very important, but given on the conditions prevailing in that particular region where these studies have been undertaken. Platyveliger, transitional stage, swimming to crawling. Pediveliger stage, now eye spot stage has developed into platyveliger stage. This platyveliger stage shows transitional stage from swimming to crawling. Earlier it was swimming stage, but cilia, flagella, everything was lost, shell developed. So, they have from swimming, they have shifted to crawling movement. Now, in the platyveliger stage, velum and foot develop. Time on day 18. Then plantigrade. Here, foot becomes functional. Since in the previous slide, if you remember, they were this platyveliger stage was representing transitional stage, where from swimming they started crawling, but now foot becomes functional. Foot developed and became functional in plantigrade. Byssal gland active and secretes byssus threads, which is very necessary for attachment. So, in plantigrade stage, the provision for attachment to the hard surface is there, and this is on day 20. So these timelines and the characteristics with timelines becomes very important to remember. Then spat, rapid shell growth everywhere except umbo region. So labial palps, additional gill filaments and byssus threads develop. Extension of anterior and posterior ear, and this occurs on day 24. Larval development of Pinctada fucata, it has been shown here. You can have an overview. Early umbonal larvae with calcified shell. In A, you can see early umbonal larvae with calcified shell. In B, you can see umbonal with prodysoconch 1 and 2 in case of B. Then umbone stage, the C representing umbone stage, D later umbone stage, and it is followed by E representing ACC, amorphous calcium carbonate, and inner prismatic layer, that is represented in E. Then we have D stage hinge, which is represented in F. D stage hinge of a single valve and a section of broken shell shown magnified in D. And it is followed by this G stage, where umbonal shell can be seen, H eye spot stage, and the last one, that is spat stage. So that's all in the biology of Pinctada fucata. In the next lecture, we shall be discussing about the culture method. Thank you.